speaker of this session is Dr. Sapna Katoch or Dr. Deepak Kumar from GGN Science College.
So they are not there in the meeting.
They are not in the meeting.
No, sir.
So let's move to the next one. And the next one is Dr. Nirupama,
Yes,
Assistant
sir.
Professor in Chemistry, Dr. Rajinder Singh. And who is speaking?
Sir, myself, Dr. Nirupama.
Nirupama is
I'm
speaking. Great, great.
Yes, sir.
So Nirupama, please share your slide uh,
Okay, sir.
and start your lecture. And we limit within 10 minutes of time.
Okay, sir. My screen is visible to all of you.
Yeah, it's visible, it's visible, it's visible.
Okay.
You
Good
can,
afternoon,
you can go ahead.
Pandey sir, and my warm good afternoon to all the organizer of this conference. Today, I will present my paper on the topic adducts of this O-isopropyl diethyl copper 2 with nitrogen donor ligands and their biological activity. So, I am giving the brief introduction about the xanthates, or they are also called O-alkyl diethyl. They are they have been known since 1815, and they were first prepared by chemists xanthos, owing to the yellow color of the lead xanthate. Xanthates of the alkyl metal sodium and potassium in particular are widely used employed as a reagent in the selective separation. They are the important classes of 1,1 diethylate family among the dithiocarbamates, dithiocarbоxylates, dithiophosphates, and other related phosphor 1,2 dialate ligands. Xanthates are actually the reaction product of carbon disulfide, an alcohol, and an alkali. Xanthate ion ROCO2 negative display a wide variety of coordination pattern as shown below. They may coordinate as to metal as in the SS mode or in the SO mode. Apart from these two coordinating modes, xanthate can also bind to the metal in following ways as shown in the figure: in a monodentate ligand, in isobidentate, an isobidentate fashion, or it may also involve oxygen during its bonding. Alkali metal xanthates are pale yellow solids in the pure form and have a faint odor. When kept in atmosphere, a deep yellow color and rather unpleasant odor resembling that of the carbon disulfide is seen, which is due to the slow decomposition of xanthate in the atmosphere, and the product of decomposition, especially organosulfur compounds, cause the disagreeable odor. Xanthates of alkaline earth metal are also known, and they are again prepared by the reaction of alcohol, carbon disulfide, and the metal hydroxide in the stoichiometric ratio of 2:2:1 with elimination of water. The complexes of monoanionic diethylcarbomates and xanthate ligands with transition metal, including nickel, continue to attract the attention due to their use as the precursor for the preparation of solid state metal sulfide materials. Again, xanthates have the wide application. They are mainly used as the fungicides, pesticides, vulcanization accelerators, flotation agents, corrosion inhibitors, high pressure lubricants, and they are quietly used in the therapy of HIV infection. Metallic xanthates are well known reagents in the flotation of m- minerals, transition metals such as copper and nickel, and in the separation of quantitative determination of large number of cations. Cadmium methyl xanthates are used as the precursor for synthesis of cadmium sulfide nanoparticles capped with the trinanooctyl phosphine oxides. Xanthates found a practical application as additive to the lubricating oil, antioxidant for the polyolefins. They are also used effectively in stabilizing the soil against the filtration and the deformation. For example, the cellulose xanthate at the concentration of 1%, 0.1% in the soil reduces the soil erosion, but when it is present in the high concentration, it binds soil to improve their mechanical strength. Then the experimental method used for the preparation of different adducts involves three-step preparation of the potassium salt, preparation of their parent compound, and the preparation of adducts. The potassium salt of isopropyl diethyl are prepared by the standard method in which we can use 4.2 gram of potassium hydroxide we take 4.2 gram of potassium hydroxide in round Barton flask, and we add about 0.26 moles of isopropyl alcohol. The reaction mixture was heated under the reflux for 1 hour, and the contents were then cooled, and liquid from the reaction mixture was decanted off into another dry conical flask. To this flask, we add 0.05 moles of carbon disulfide, and the flasks were again filtered. Contents were filtered on an sintered glass funnel at the pump and washed with the ether in order to dry the product. In the second step, the attempts were made to synthesize the complexes of this alkyl dithicarbonate of copper 2 by mixing the aqueous solution of potassium salt of alkyl dithicarbonate with cupric chloride. Though initially brown color precipitates of the bis O-alkyl copper 2 were obtained, but these brown color precipitates on tanning get reduced to the yellow color precipitate, which corresponds to the stable bis O-alkyl carbonato copper 1. So all the attempts to isolate the stable copper 2 adducts were not possible. So rather than proceeding with the normal method, if we proceed with the parent compound when the reactant in a manner when the reactant, that is cupric chloride dihydrate, potassium salt of O-alkyl dithicarbonate, and nitrogen donor were mixed in the ratio of 1:2:1 in the acetone, the formation of stable product of this O-alkyl dithicarbonate of copper was observed, as confirmed by the various, and these products are confirmed by the various physicochemical techniques and the spectroscopic studies. Then, after the preparation of parent compound, the adducts were prepared by dissolving the parent compound in acetone, followed by the addition of the stable nitrogen donor complex. All these complexes I have prepared. In this paper, I am presenting one series in which I have used 8 nitrogen donors. So, results and discussion. The preliminary investigation of all the adducts prepared by using this O-isopropyl dithicarbonate of copper 2 with the different nitrogen donors which I have used, like 2-bromo, 3-bromopyridine, 4-acetylpyridine, 3-hydroxypyridine, 2-methoxypyridine, 2-amino, 3-methylpyridine, 2-amino, 4-methylpyridine, and 2-amino, 5-methylpyridine. The adducts formed are insoluble in common organic solvents such as acetone, 1,4-dioxane, nitrobenzene, ethanol, methanol, etc. However, they are soluble in methyl sulfoxide, dimethylformamide, and the parent compounds. It has been observed that the parent compounds are less soluble and they get reduced to corresponding copper 1 dicarbonate. But once the adducts are formed, they are highly stable, and on the analytic data of the adduct shows that the complexes they are quite stable and their yield is also quite good. And all these complexes, adducts, which are prepared by using the nitrogen donors, they are characterized by the various physicochemical techniques, such as molar conductance measurement. The molar conductance measurement, the values of molar conductance measurements fall in the range of 10.84 to 24.863 ohm inverse, mole inverse, centimeter square. These values are lower than the values expected for any univalent electrolyte in their covalent behavior, suggesting that these complexes are neutral and non-ionic in character. The molar conductance data is also shown in the table. Similarly, these complexes are also characterized by the magnetic measurement. And since copper 1 can be value of 1, the copper has the ground state complexes. We have the configuration E4 and T, which is 5. And the magnetic values are in the range of 1.72 to 1.82 Bohr magneton for the complexes, which shows that these complexes have an unpaired electron, and the complexes prepared in the adducts prepared in this paper, reported in this paper, they are square pyramidal in nature, and they exhibit the square pyramidal character. So this table shows the values of Then the third study which I have used for the characterization of the complex is the infrared study. The infrared spectra of the adduct, these adducts were recorded in the range of 4,000 to 200 centimeters. And the important infrared peak. Yes, sir. Yes, sir.
Nirupma,
Yes, sir.
Dr. Nirupma, can you finish
Yes, sir.
fast?
Yes, sir.
Yeah, please finish
Okay,
fast.
sir. Okay, sir.
Please do fast.
Okay, sir. Okay. So in
Okay.
these, the infrared studies shows that the complexes, the vibrational bands which are observed for these complexes are in accordance with the data reported in the literature as per the theoretical values. So these are the values of different vibrations observed for these adults. And the main difference in the IS spectra shows that the value for CS bond instead of the CWS bond as present in the carbon disulfide, there is decrease in the value of vibrational frequency, which shows that the the xanthate ligand is coordinated to the metal through the S. The electronic spectra of the complex is recorded in the range 12,500 centimeter inverse to 40,000 centimeter inverse. Again, since the geometry of the complex reported adducts are square pyramidal in nature, so the different type of transitions which are observed in this case are from dx y to dx square minus y dx square y square, dz square to dx square dx square y square, and dxy to yz to dx square minus y square. And one important thing which was observed in this complexes was that the five coordinated nature of the copper 2 complex will experience a John Teller pentagonal distortion, which with the fourth short metal ligand bond in the XY plane. So, in the spectra, in the present work, we observe the intense band corresponding to DG transition as observed in most of the copper 3 complexes in the range 1500 500 to 17,600 centimeter inverse. The main absorption band around 16,000 is assigned centimeter inverse is assigned to dx z and dy z to dx2 transition, and the weak shoulder is associated with the transition due to dz square to dx square minus y square transition. And the third possible transition depends upon the extent of the axial ligand metal interaction and the appearance of single band of high intensity suggestive square pyramidal geometry around the central copper metal line. This is the electrospecstroskopic data, electronic spectroscopic data of the complex, and these complexes are further characterized by the ESR studies. The ESR studies, since the copper has a spin of 1 by 2, so we have the two splitting pattern according to the rule 2i plus 1. So the two peaks which were observed in the ESR spectra of the copper 2 complexes, which I have prepared, shows that the copper exists in the 2 oxidation state and not in the plus 1 oxidation state. Further, these complexes were characterized by thermogravimetric analysis, which show the gradual decomposition of this product as we increase the temperature, with the copper sulfide as the final end product. The mass spectral studies of the complex also shows that the complexes are quite stable, and the peaks corresponding to the higher MZ value can be regarded as the direct fragmentation of the molecular ion, while the peak which corresponds to the lower M by Z value are considered as the daughter nuclei. So, the mass spectra of the adduct with the 3 bromopyridine is shown in the figure, and the peak corresponding to 198.40 shows the base peak, and the further the degradation, the removal of the mass particles. This is the table which shows the decomposition of the complex as during the mass spectral study. The antifungal studies were also carried in order to study the behavior, the biological behavior of the complex, and it has been found that the technique used for studying this antifungal activity.
Please conclude now.
Okay. Okay, sir. So these are the antifungal studies which shows that the complexes are antifungal in nature, and with increase in the concentration of the complex, there is decrease in the colony size of the sunlight. That's all about my paper. Thank you so much. Thank you, Pandit sir.
Thank you, Dr. Nirupma. So, house is open for question, and I will take only one question. Seems there is no question. Thank you, Dr. Nirupma, for your nice presentation.
Thank you, sir.
Now I move forward to invite the next speaker. The next speaker, there are two names. I don't know who is going to present, Dr. Pooja Nanda and Dr. Yashpal.
Yes, sir, I will present. I will present on behalf
Yes,
of
Dr.
Pooja.
Yashpal is going to present. So please, Dr. Yashpal, share your slide immediately.
Yes.
It's fine.
In the chair, Professor Sushil K. Pandey, Rector, Riyasi campus of University of Jammu and ex-HOD Department of Chemistry. Principal of the college, Professor S.P. Saraswat. Sir, HOD Chemistry, Dr. Avinash K. Gupta. Sir, other members of the organizing committee, representatives from Cape, there is Cape some organization, if they are Cape organization, faculty members of Department of Chemistry, GCW Parade, especially organizing secretary, Dr. Gurpreet Kaur. A very warm good afternoon to all of you. Incidentally, I would like to congratulate Dr. Avinash sir for organizing this event. Since I am associated with this college and I also worked in this college for a few years, I think this is the first international conference this department is organizing since its start. You can say this department was established in 1963. So as per my knowledge, I am in chemistry for last 25 years. And as per my knowledge, this is the first international event which you are organizing. And the way you are organizing, I think that is really commendable.
Thank you, Dr. Rao.
This is all because of this. You have a blend of youth and experience.
I think
whenever you mix these two things, result will there to come. Result will always there to come. My presentation that this is I am not discussing a rocket science today, and it is not possible for me where I am posted and where I am working. It is not possible for me to discuss something for Nobel Prize or something which cannot be understood. My presentation today can be understood. I had prepared a compound that is antimony trichloride. And only one, this is a single part synthesis which I carried out. You need a small laboratory for this only. And if you decrease the cost of a compound, if you have a method with which you can decrease the cost of a compound by 50%, I think that is enough for a researcher like me to share with other colleagues and students. My presentation is dedicated to my mentor, Professor Sushil K. Pandey, who is by chance sharing this session. मिट्टी था सर, मैं मिट्टी था, सीरियसली मिट्टी था. मैं तो बुद्ध था, मैं तो आदमी आपकी वजह से बना. You are the man who monumental and metamorphosed me into a human being.
Thank you,
And
Ishpal, for
I owe myself to you, sir.
Thank you, Ishpal.
This is antimony trichloride, commonly known as butter of antimony. Am I audible to the participants?
Yes, you are audible.
Yes. My presentation, what I did is basically, I prepared antimony trichloride, and I am inspired by a paper of you, sir, which you published in 1997, co-authored by H.W. Royski from University of Göttingen, Germany, and another scientist, A. Steiner. This reaction of mine is inspired by the preparation of arsenic trichloride, which I used to use in my PhD, and even my lab mates which followed me in doing PhD under your supervision, they also use that antimony trichloride. This method is very simple. In the laboratory, in industry as well as, antimony trichloride is prepared by different methods. In this screen, you can say it can be prepared by the reaction of antimony trioxide with disulfide dichloride. It can be prepared by the reaction of antimony trioxide with chlorine gas. Very simple methods. These are simple methods and these are available in the market. In India, whole of antimony trichloride is prepared by the reaction of antimony trioxide with HCl. But one thing is very clear, since I worked with these metals, and I know how hygroscopic and how moisture sensitive these compounds are. So it will be very difficult to prepare antimony trichloride by the reaction of antimony trioxide and HCl, because water will be formed, and since compound is water sensitive and compound is highly moisture sensitive, so I don't know how they save this antimony trichloride from hydrolysis. So another method where you need chlorine gas, you need sulfur powder, you need antimony trioxide. So these methods are costly as well as time consuming. So that's why in our laboratory we used to prepare antimony trichloride, arsenic trichloride by a reaction, arsenic trioxide with thionyl chloride. This is the only reaction which I will discuss. I will take only 5 or 7 minutes more. Antimony trioxide is a ash-colored powder. You have to take antimony trioxide in a round bottom flask, preferably double necked. If you do not have a double necked round bottom flask, use a Claisen head and put that antimony trioxide, say 100 grams of antimony trioxide, and through the dropping funnel, thionyl chloride is added. Thionyl chloride is very cheap. Commercial thionyl chloride is only 32 rupees per liter. Antimony trioxide is a bit costly, but thionyl chloride, and you need not to use pure thionyl chloride. You can use commercial thionyl chloride. So SOCl2 is acting. What is the beauty of this reaction? The beauty of this reaction is there is no solvent. We are using SOCl2 as a solvent as well as a reactant. So when we put thionyl chloride in antimony trioxide, sulfur dioxide gas will be evolved. And if reaction start, you just add 50 milliliter of thionyl chloride, and after addition of 50 ml, the reaction will start. There is some setup. This reaction will be carried out under Saline conditions, under nitrogen atmosphere. Atmosphere must be dry, since these compounds are, antimony trichloride is highly moisture sensitive, hygroscopic as well. So reaction is carried out on a magnetic stirrer. A round bottom flask is fitted, then thionyl chloride is added, and once the formation or evolution of sulfur dioxide is there, it means reaction started. So you continue adding thionyl chloride slowly with the help of that dropping funnel till as bubbles of SL2. Put a bubbler next and do something for the removal of sulfur dioxide. Use a pipe and exit that pipe to the outside of your laboratory, so that will not harm you. And stir the reaction mixture for 30 minutes or 45 minutes. Once evolution of sulfur dioxide is stopped, remove the solvent, that is SOCl2, which is acting as a solvent as well as a reagent here. Remove solvent SOCl2, dry the compound, you will get crystal crystals of antimony trichloride. And this method, why this method is useful? Because our byproduct is a gas, but that is a poisonous gas. Even antimony trichloride is poisonous. So what we will do for this sulfur dioxide? I have an idea for utilizing this sulfur dioxide, but I will not share here. So later on, after doing some work, I will definitely publish it somewhere. I'm trying for a patent of this reaction, so that it can be used by industry in India. This is a method which I shared. Now, what is the price of antimony trichloride in the country? If you require antimony trichloride, 1 kilogram packing is of 5,000 rupees. But this method, I calculated all the costs, I check all the cost, and you can purchase it by 2700, 2800. So cost is almost 50% decreased. So it is used in many antimalarial trichloride has many uses. It is used as in the detection of vitamin A. One very beautiful application which I used, this antimalarial trichloride we used I used in my childhood. It is used basically for the removal of horns of calves instead of cut them. So we used to remove the horns of the calves. Either you have to cut them or you have to remove. So this is recently used to, but this is poisonous. This is a reference. This is the paper which I used as a reference from where I get this an idea. Thank you, sir. I would like to acknowledge my college, government Padma Shri Padma Sachadev Government PG College for Women, Gandhinagar. There is a lab, interdisciplinary research lab under hub and spoke model. We have some facility like magnetized stirrer, heating mental. They give us some money. The lab is set up, and I repeat my reaction there and prepare this antimony trichloride. Before presenting, I prepared it twice. So did element analysis of antimony and trichloride for. Thank you, sir. Thank you for giving me an opportunity. Thank you.
So thank you, Dr. Ishpal. First of all, let me say I am overwhelmed by your kind word, the respect which you have paid to me. But on the other side, I feel rather satisfied in a way that you have used one of my publications to do something innovative, and that's a good thing. And well, you did not told anything about the SO2 what you are going to do. You have kept it.
Yes, sir, that is a secret for the time being.
You have kept it secret.
Yes, sir.
But definitely nowadays it is very important when you are conducting saturation that must be very much environmentally friendly reaction.
Sir, I will not allow even a iota of sulfur dioxide to the atmosphere. Let me promise you.
That's okay. That's okay.
Yes.
So
and I will not going to lose any sulfur dioxide. Sorry, thionyl chloride even. I had used thionyl chloride for two purposes here, as a solvent as well as a reagent. So there will be no pollution from my side.
And secondly, my advice to you is that don't go for patent, go for publication. So that will help the young students and the scientific community around the world. So commercially, there is no issue of money. The money is issue in case of the research fraternity. So better idea would be to publish. So everybody should benefit about this new method of the synthesis of antimony trichloride. Okay, now session is open for questions. One question, please.
Thank you.
So there is no question, and once again I say thank you to Dr. Ishpal.
Sir, I would like to say one more thing, sir.
I think
before end. So today is the death anniversary of our great Prime Minister Shri Lal Bahadur Shastri ji.
Yes.
So I would like the chairperson to say few the participants to have to pray for the departed soul, or you can say pray for the great leader.
I don't know whether this protocol of the conference allows to do it or not. I think convener can be the better person for this.
Okay, sir. Thank you, sir. Thank you, sir.
So now move to the last speaker, and she is Pavandeep Kaur. Pavandeep, are you ready now? Pavandeep Kaur, please. Pavandeep Kaur, please.
So she is there in the meeting. Pavandeep, please unmute yourself and speak. Can you hear us? Yeah, she is presenting,
Yeah,
sir.
she is on the task. Great.
I request all the speakers to please restrict to the time limit.
Yes, 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 yes.
Yes, sir, I'm going to present now.
Yeah, okay. So Pavandeep Kaur, research scholar from the Department of Chemistry, University of Kashmir, is going to speak on interfacially engineered microemulsion loaded biopolymeric hydrogel capsules, that is a novel food grade delivery vehicle for stabilization and protection of long chain polyunsaturated fatty acids against peroxidation. So now, Pavandeep, please start and go to the slide show mode.
Okay, sir. Sir, is it okay now?
Oh, it's now wonderful, Pavandeep. So,
Okay.
you know,
Ji, sir.
after waiting for a time, you have a very good result.
Thank you, sir. Sir, I am a student of Jammu University when you were HOD over there.
Oh, great, great.
Ji.
Great. God bless you.
Thank you, sir. Okay, good afternoon, everyone. This is Pavandeep Kaur, a research scholar from the Department of Chemistry, University of Kashmir, working under the supervision of Dr.
Request to the audience, please mute the mic. Yes, continue, please.
Okay, good afternoon, everyone. This is Pavandeep Kaur, a research scholar from the Department of Chemistry, University of Kashmir. I'm working under the supervision of Dr. Ajaz Ahmed Dar, and I'm here to present one of my research works entitled Interfacially Engineered Microemulsion Loaded Biopolymeric Hydrogel Capsules, a novel food-grade delivery vehicle for the stabilization and protection of long-chain polyunsaturated fatty acids against peroxidation. Let us have a brief introduction about long-chain polyunsaturated fatty acids. Long-chain polyunsaturated fatty acids are those fatty acids which contain 18 or more than 18 carbon atoms. Their main sources include eggs, nuts, vegetable oils, etc. They are basically classified into two families, that is omega-3 and omega-6. Whether the given LC-PUFA belongs to omega-3 or omega-6 family, it depends upon the position of first double bond when numbering starts from the non-terminal COOH from the non-COOH terminal. For example, this is linoleic acid. It has two terminals. One terminal contains COOH group and the other contains CH3 group. Now, if we start numbering from this terminal, which is a non-COOH terminal, we can see that the double bond first double bond appears at 1, 2, 3, 4, 5, and then 6. That is at 6th position, first double bond appears. So it is omega-6 LC-PUFA. Similarly, this is Icosa pentanoic acid. In case of this, if we start numbering from the non-COH terminal, we can see that the first double bond appears at 1, 2, and 3. That is third position. That means it is omega-3 LC-PUFA. Similarly, linolenic acid is another LC-PUFA, which is omega-6, and docosahexaenoic is omega-3 LC-PUFA. As far as their therapeutic activities are concerned, they cover a vast arena, which includes the developmental bone, brain, and body. Additionally, they act as the anti-cancerous and anti-diabetic agents as well. But on exposure to high temperature, heat, or oxygen, these LC-PUFAs undergo rapid oxidation, which may lead to the rancidity and production of off flavors. And ultimately, the result is toxic end products, which may badly affect our health. From time to time, different formulations were developed to control their oxidation. Some of the important formulations include micelles, nanocrystals, liposomes, hydrogels, emulsions, etc. Although all of these served for years together for the proper encapsulation and delivery of bioactive molecules, but some of them are associated with serious drawbacks. For example, if we talk about micelles, they undergo thermodynamic instability. If we talk about these liposomes, they undergo leakage of encapsulated material. Some undergo burst release, and so on. As I am working in soft matter research group, so we decided to club both the emulsion and hydrogels in a single unit so as to get the dual benefits of both the delivery systems by overcoming all the possible limitations or drawbacks. Among emulsions, we chose microemulsion. Why? Because they consist of very small droplets which are thermodynamically stable. They are clear and have transparent appearance. They are known to solubilize the hydrophobic bioactives without compromising their bioavailability, shelf life, taste, or odor. On the other hand, if we talk about hydrogels, as it was also discussed by so many presenters before, but here what we are doing, we are using both microemulsion and hydrogel in a single entity. Here these hydrogels, these are the three-dimensional polymeric network which are known to retain large amount of biological fluids, and they have good mechanical strength, they have good swelling power, and they have high porosity, which aids them to present a sustained drug release. On combining both these entities, we get a single wonderful assembly which is known as microemulsion loaded by polymeric hydrogels, which is of course a novel discovery in the sustainable food system. What we did in our work, we actually prepared 10 microemulsion formulations, as I have numbered them from F1 to F10. What we did, we use cis-linoleic acid, which is an LC-PUFA, as an oil phase in this emulsion. The most interesting thing is that in order to make these emulsions, we used a range of surfactants. For example, we use non-ionic surfactant, we use anionic surfactant, we use zwitterionic surfactants as well. It is so because the oxidative degradation of lipids in emulsions is believed to be initiated at the interface. And once the interface is protected, it will protect the encapsulated LC-PUFA. As you can see from this table, the total weight percent of cis-linoleic acid was fixed at 1%, whereas that of surfactant was fixed at 10%. Here T20, it stands for tween-20, it is a non-ionic surfactant. LS stands for lauryl sarcosinate, which is an anionic surfactant, and DPS stands for 3-NN dimethyl myristyl ammonio-propane sulfonate, which is a zwitterionic surfactant. All these microemulsions were clear and transparent. Their particle size was characterized by DLS studies, that is dynamic light scattering studies. From this graph, we can say that all these 10 formulations, they have the droplet size of microemulsion less than 100 nanometers. But those which were stabilized by anionic surfactant, they have comparatively greater or higher size as compared to those which were formed by non-ionic or zwitterionic surfactants. The most probable reason seems to be the electrostatic repulsion between the negatively charged interface and O2 negative ion of aqueous phase, which increases the droplet size of anionic surfactant stabilized microemulsion. If we talk about the zeta potential tells us about the charge of the droplet. We can see that all these microemulsions, they have a size, less than 100 nanometers, but they have the droplet size in negative terms. That means it is an ideal value for the microemulsions. And as expected, those formulations which were stabilized by anionic surfactant, they have more negative values than those formulations which are formed by non-ionic or zwitterionic surfactants. Moreover, after 3 months, when we monitored these 10 microemulsions, they were still transparent with negligible increase in droplet size. It means that the microemulsions which we formed were very excellent in terms of their stability. Now, for monitoring their oxidative stability, we measured the formation of conjugated dienes, which are the primary oxidation product of LC-PUFAs. For this, a very potential antioxidant quercetin was used. Here you can see we made 10 formulations of microemulsions which didn't contain quercetin and 10 formulations which contained quercetin. When we analyzed the formation of conjugated dienes, as you can see in both these graphs, those formulations which didn't contain quercetin, the amount of conjugate diene formation was considerably large, whereas those formulations which contained quercetin, they presented much decline in the formation of conjugated dienes. It therefore revealed that LC-PUFAs loaded into the microemulsion for protecting them against the oxidative degradation. Furthermore, we can see that this is the graph, this is the formulation for tween-20 in absence of quercetin, and this is the formulation for tween-20 in presence of quercetin. It means that tween-20 was not so much good for controlling the process of oxidation. It was so because the long chain of hydrophobic tail of twin 20, which lies in close vicinity of oil droplets, and it itself gets involved in promoting the oxidation of underlying lipid droplets. On the other hand, in other formulations where twin 20 was replaced by a small fraction of twin 20 was replaced by anionic or zwitterionic surfactant, a very large decline in the oxidative tendency was observed. It may be because of the presence of micelles of anionic or zwitterionic surfactant in aqueous phase, which would keep away the oxidation causing agents to approach the interface. Next, we incorporated, I told you priorly that we use both microemulsion and polymers in a single assembly. What we did, we used these microemulsions and incorporated them into the hydrogel. And among hydrogels, we chose alginate, and by using calcium chloride, we made spherically shaped these alginate capsules, whose size ranged from 2.0 to 2.30 millimeters. But we can see here that we made 10 microemulsion formulations, but here I am representing the size of only 6 capsules. It is so because there were some microemulsions which contained highest amount of negatively charged surfactants. What they did, they got precipitated when they come in contact with the calcium chloride ion. So we were left with only these 6 formulations which survived with those formation of capsules. Now, when their IR spectrum was observed, we can see that there was a change in the position and intensities of the peaks, which means that a mere change in the interface of the microemulsion can bring out huge changes in their interactions with the coating of the alginate. Now, the gel strength of the capsules is another important factor which can be seen from these amplitude sweep tests which we carried out on the instrument known as rheometer. It is basically a plot of G prime and G double prime versus strain. Now, what is G prime and G double prime? G prime represents storage modulus, whereas G double prime represents loss modulus. Storage modulus means which depicts the gel-like character, and G double prime, that is loss modulus, represents the sol-like character. The point where G prime and G double prime cross each other is known as crossover point. Greater the crossover point, greater will be the gel strength. Among all these 6 formulations, we can see that in this formulation where twin 20 was used as 2% and DPS, which is a zwitterionic surfactant, was used as 8%, the maximum crossover point is detected. It means out of all these 6 capsules, this particular formulation, it was very strong in terms of its gel strength as depicted from this figure. Now, whether these capsules, how much self-healing tendency they have, it was carried out by using the thixotropic test, which is also carried out on rheometer. It is a plot of G prime and G double prime versus time. Now, what does these plots means? Here the black lines, they represent the G prime, that is the gel-like character, and the red line, they represent the sol-like character. It is known as loss modulus. You can see here when no strain was applied, the capsules were in the gel form. That's why G prime and G double prime, they have very large values. As soon as we applied strain on it, the entire structure ruptured. You can see here that the black line moved from this to this place and the red line moved from this to this place. But as soon as the strain was released, again it got back to its original place. That means from these figures, in all these figures, we can say that all these capsules, they have the tendency of self-healing. This self-healing tendency is known as thixotropic effect. And what we can conclude from this, we can conclude that all these capsules, they can be used for the storage purpose without any fear of leakage. Sometimes while storing or while traveling, we can got some pressure on these capsules, but they will not undergo any type of leakage of the encapsulated material. That means these are good for the storage purposes. From this figure, we can see that it is a plot of viscosity versus shear rate. It is also another test which can be carried out in rheometer. From this figure, it can be seen that on applying the shear, their viscosity decreases. These are the viscosities of the capsules which goes on decreasing on increasing the shear rate, and this decrease in the shear, decrease in the viscosity with increase in the shear rate is known as non-Newtonian shear thinning behavior. And this behavior is very important, which enables.
Pawandeep,
Ji sir.
can you conclude very fast?
Yes, sir. I have only one slide after it.
Okay, great.
हाँ जी, sir. So this is the non-Newtonian shear thinning behavior, and it is this behavior which enables them to fit for oral delivery. Why is it so? Because if we take any capsule or any medicine inside our body, and on chewing it, its viscosity should decrease. Now only then it can be digested. It is known as the shear thinning behavior. If it would have increased, then it would be unable for us to digest it or to get the benefits from it. From this graph, we can see that on immersing these capsules into the saline media, which is the having the physiological pH of our body fluids, the capsules swelled up, but they didn't dissolve. That is another good factor. And the third graph, it represents the percent RSA, that is percent radical scavenging activity. I told you initially that we used quercetin as an antioxidant in order to protect our LCP5 against oxidation. Now what we did, if we use it for scavenging DBPH free radicals, we can observe that different type of capsules represented different ways and different trends to scavenge the free radicals. That means in a single entity, we incorporated LCPUFA, we protected it, and by making its capsules, when we released quercetin from it, it did wonderful role in scavenging the free radicals as well. Now, in the conclusion, I would like to say, in nutshell, when microemulsion with along with the linoleic acid as oil and quercetin as antioxidant are formed, they can protect the loaded LCPFAs against oxidative degradation. The capsules marked wonderful gel strength, as we can see from the graphs of the rheometer, and the non-Newtonian shear thinning behavior with no leakage at all. Additionally, both quercetin and linoleic acid are still present in their biologically active form when loaded inside microemulsion alginate capsules. Thank you.
So thank you, Pavandeep.
Ji, sir.
It's a really wonderful presentation.
Thank you, sir.
Really, I appreciate it. Is the very good, very well organized, and very well presented.
Thank you, sir.
Wonderful,
Thank you
wonderful.
so much. Thank you.
Of course, the work is also of very high quality,
Ji.
and the presentation and slides and the synchronization is
Ji.
really needs really an appreciation from
Ji.
for you. I appreciate personally.
Okay,
Very
thank you so much, sir. That means a lot. If I'm
Very-
presenting it before you, that actually means a lot.
Yeah,
But you
that's
are
give
praising,
me
that means much.
that give me a lot of satisfaction.
Ji, okay, thank you,
So,
sir.
so house is open for one question from Pavandeep.
Ji.
Any question?